Bismillahirrahmanirrahim. Ladies and gentlemen, Assalamu alaikum and a good morning to all of you. Hello and welcome to you to the 3-day international webinar on water, health and parasites. Today is the second day for this international webinar, that is technical session second, wherein today we have with us our guest, Dr. Samindra Sarchan, and he is an assistant professor from Department of Global Environmental Health Science, Tulane University, USA. Before we directly go to his talk that's titled Wastewater-Based Epidemiology, Current Challenges and Limitations, I would like to introduce our today's speaker, Dr. Samindra. Dr. Samindra Sherchan is an environmental health microbiologist and has served as a subject matter expert in the areas of on-site water treatment and water quality for the National Environmental Health Association. He is currently working as an assistant professor in Tulane University, USA, and teaches undergraduate and graduate courses in microbiology, infectious disease, environmental health concepts, environment and human health, water quality management, WASH, and global health, public health microbiology. Prior to joining the faculty at Tulane University, Dr. Samindra was an assistant professor at California State University, Fresno. Dr. Samindra received his BS and MS in Biology and Environmental Sciences from Georgia College and his PhD in Soil, Water, and Environmental Sciences from the University of Arizona, USA. Dr. Samindra has specific research interests include water quality, emerging contaminants of concern, infectious disease, mechanism of pathogen inactivation, environmental virology, advanced detection techniques for emerging contaminants, bioinformatics, microbial source tracking, metagenomics, risk assessment, water treatment, water reuse, and challenges associated with safe drinking water, sanitation, and hygiene. Dr. Samindra has contributed more than 60 publications and is a registered environmental health specialist. Due to his immense contribution in educating the public, he has received the awards like Technical Advisory Online Sewage Treatment NEHA Award in 2014, Best Poster Award in 29th Annual Water Resource Symposium, and also WF Award, Water Hero Award in 2020. I think with these comments, remarks on his professional competency and keeping in the theme of this, that's water, health, and hygiene, I am confident that this will be of immense importance and relevance to our participants who have actually logged in right now for this international webinar. I think with these remarks on his actually contribution, I would like to actually invite our today's speaker, Dr. Samindra Sherchan, to kindly share his presentation. Please, Dr. Samindra.
Hello, everyone. It says host to disabled screen sharing. All right, so can you guys see the presentation?
Yes, it is coming. Your presentation is coming. You can go ahead, please.
All right, thank you so much, Dr. Bhatt. So today I will be talking about wastewater-based epidemiology, current situation, challenges, and limitations. Before I start, I would like to thank my lab members, some of my collaborators in the US and outside the US in Australia, Japan, and Nepal and India. So my funding agencies here from the US National Institute of Health, National Institute of Allergy and Infectious Diseases, and National Science Foundation. I would like to thank my graduate students who were really a part of this project looking at SARS-CoV-2, the virus that's causing this pandemic, COVID-19. And then past year, they've been working very hard in the lab. So here's my outline, what I'm going to be talking about today. First, I'm going to talk about what is as an early warning system to monitor COVID-19 prevalence in communities, and it can be used as an early warning tool for preventing potential outbreaks. What are some ongoing research opportunities in this field, and some of the communications and collaborations for future? So wastewater testing, it's not a new study, because we know that we've been monitoring sewage or waste water for other pathogenic viruses like poliovirus. So this is kind of first article back in 1941 that looked at wastewater for the poliovirus. So as we know, that poliovirus was a problem back in 1939 and 1940s. During this time, without a vaccine, a lot of people that were infected with polio, and people who were working in wastewater sewage surveillance, they were monitoring this virus to look at the outbreaks. So what they found was, periodic sampling of wastewater for poliovirus. It had a very good advantage for public health officials to mitigate and combat this disease. Uh, and after several years, they invented the vaccine in 1954. But we are still seeing the wild strains of polio outbreak in different countries like Israel, Afghanistan, Pakistan. So what they are doing is using wastewater testing, environmental surveillance to monitor polio. So it's been a long time, almost 60, 70 years since polio, but there are still some countries with this problem. And to eradicate polio, we need to have a robust surveillance program. And there are some challenges with clinical testing, with asymptomatic and pre-symptomatic individuals setting the virus. So environmental surveillance using wastewater for detecting pathogens, it's a very key for eradicating infectious disease. So this is just an example, and there are other viruses in wastewater, as you can see, mostly enteric viruses like rotavirus. So that's the number one virus that cause gastrointestinal disease below 5 years of old. Norovirus, adenovirus, which is DNA virus, astrovirus, reovirus, influenza, so human HIV virus. So these are some of the virus that can be detected in wastewater frequently. So just to go back to last year around this time when there was a pandemic, we did not know a lot about this pandemic. Most of us thought this was, due to person transmission. But there came few papers, March 2020, where they were looking at SARS-CoV-2 virus in stool samples. Confirming that you can detect the viral RNA in stool samples or feces of infected individuals. And in some countries, in some of these articles, it was very fascinating to see you can even detect the virus in stool, even the symptoms or respiratory symptoms are gone. And then in China, we had isolation of SARS-CoV-2 virus from a stool specimen. So with a lot of these articles last year around March 2020, a lot of groups started looking at wastewater surveillance or wastewater-based epidemiological studies. So what is this is mostly we are collecting wastewater samples, looking at SARS-CoV-2 RNA. So there are a few early reports, showing the presence of SARS-CoV-2 in wastewater. And we can use this as an early warning tool because we can identify what's the trend of COVID-19, cases in the community. And it can also complement the clinical testing and see whether you need more strict regulations, public health actions. And it can capture both symptomatic and asymptomatic infection. Unlike clinical testing, because you have a few limitations, so like asymptomatic, they might not, show any symptoms. But with the wastewater, if they are setting the virus in the stool, you can have pool testing approach, and it can also overcome clinical testing, like countries where you do not have resources to do massive clinical testing. Wastewater testing can provide an alternative to monitor COVID-19. So back in 2020, April, we started looking at wastewater samples. We published the first report on the detection of SARS-CoV-2 RNA in wastewater. So what we did was we collected wastewater composite and grab samples, and then we brought them into lab and processed using ultrafiltration and electronegative membrane filtration. We extracted the RNA from the concentrates and used the quantitative PCR method to look at the virus. So we were able to detect, few positives in the influent, which is, the raw sewage that's coming in the wastewater treatment plant. We did not detect any positives in the secondary and effluent. So this was the first report kind of utilizing wastewater testing to monitor COVID-19 in communities. So this slide right here kind of represents the overall, approach. What can we do to mitigate and combat COVID-19 using wastewater testing? So first, you have to consider which sampling point you want to collect samples from. Most studies, they are looking at raw sewage or influent, as you can see here, because that's where majority of waterborne pathogens might be. So you can collect the influent wastewater, but there are some studies that can also look at the solids, primary solids in the sludge line. That's after the primary, secondary treatment. That can also give you some indication of the SARS-CoV-2 RNA. And then if you want to look at the fate of SARS-CoV-2 in the wastewater and the aquatic environment, we can also collect secondary, tertiary, and the final treated effluent. And then for virus, because they might be lower in concentration but highly, infectious, you need to concentrate them. So you have to use any of these concentration methods. So there are several of them, like ultrafiltration, centrifugal ultrafiltration, precipitation with polyethylene glycol, ultracentrifugation, and virus adsorption elution. So all of these methods help to kind of concentrate the virus, and then you use you extract the nucleic acid, and then we use some controls and look at the polymerase chain reaction and also confirm it by sequencing to make sure that the virus you are detecting is SARS-CoV-2 and not false positive. And there have been a lot of successful stories using, wastewater testing to mitigate COVID-19, especially in targeted areas like university dorms, dormitories, prisons, senior living facilities. This were like high risk areas where you can implement wastewater testing and complement clinical testing and mitigate COVID-19 outbreak. So this is one of the successful stories we used at the university dorm back in November, August 2020. We collected wastewater grab samples coming out of the dorm in the morning and detected SARS-CoV-2 RNA. Following that, we monitor COVID-19 in the all students population using antigen testing, clinical testing. And people who were asymptomatic, they were removed from that dorm. So that kind of prevented potential outbreaks. So There are a lot of other successful stories. So this is the one we published recently using COVID-19. It was highly, published in news, as you can see here, that wastewater testing can help save COVID-19 outbreak in the university. So we have done a lot of work in high targeted areas. And there are other groups looking at SARS-CoV-2 RNA and then correlating it with COVID-19 cases. So as you can see, this is some of the early studies done in different states in 2 different wastewater treatment plants. So right here, as you can see, the coronavirus concentration, that's the number of particles per liter, and then new cases in a day. So whenever there is an increase in the COVID-19 cases, you can actually see that the COVID or SARS-CoV-2 RNA levels also go up during this time. And a lot of other treatment plants also showed similar kind of trends where, the SARS-CoV-2 RNA went up after you have COVID-19 cases, and this is what the early warning can be utilized, giving you warning that COVID-19 cases might be increasing in upcoming week. So the SARS-CoV-2 testing in wastewater is actually useful for public health officials. And this is another early study done in the Netherlands where they were able to correlate SARS-CoV-2 RNA and then the reported COVID-19 hospitalizations. So this was highly cited and one of the early studies looking at correlating SARS-CoV-2 in wastewater and COVID-19 prevalence. So after that, we have published other articles, mostly focusing on SARS-CoV-2 RNA, like for example, how long does this viral RNA survive in wastewater? So we looked at decay of SARS-CoV-2 and surrogate virus in untreated wastewater. So these are a lot of implication because most research groups, they are collecting wastewater samples when it arrives at the treatment plant, but from the people who said the virus, and by the time you wait to collect it at the treatment plant, the virus might have been degraded. So this study is very important, suggesting what's the role of the temperature. So right here, we looked at, different metrics, tap water, untreated water, and autoclave wastewater, and incubated in different temperatures, 4 degree, 15 degree, 25, and 37. So as you can see, the SARS-CoV-2 RNA persists longer at 4 degrees Celsius. So up to 35 days, we were able to detect SARS-CoV-2 RNA in untreated wastewater. So as you can see, SARS-CoV-2 and murine hepatitis virus, the decay was very similar. So we can use murine hepatitis as a suitable in a surrogate to process control the SARS-CoV-2 RNA. So after this study, it was kind of confirmed that the virus can actually survive at 4 degree at lower temperatures. So there are different data that's needed for successful implementation of wastewater-based epidemiology. So first you need the concentration of the virus in the wastewater, which is concentration gene copies per liter. And then you can also use wastewater flow rate. We need to have feces production rate, how much virus is said by the infected individuals, fecal setting rate. So with all this information, you can actually calculate the number of infected individuals in the community. But there are some bottlenecks, limitations, because there are a lot of research gaps, knowing what's the fecal setting rate, because different people, individuals, they might set the virus differently. So some people, they might set the virus just for a few hours versus there might be super setters in the community. So we do not know what's the range, amount, duration that's, individual infected or asymptomatic individuals said the virus. So that's why it's kind of limited right now. Like we cannot calculate the number of infected individuals, accurately. So estimates, they are not exact, setting rate. So a few groups have used estimated setting rate to calculate the number of infected individuals and come up with the modeling aspect to complement other health data. And also there is no standard method for processing samples. So as I showed you guys, there are like tons of different methods you can use to detect SARS-CoV-2. So some groups are using ultrafiltration, some groups are using the precipitation method. So without a uniform method, the results vary in different groups and in different countries. So there is very difficult comparison without a standard method. There's a lot of variability across samples, so correlation between model estimate and actual infection, they are still being conducted. So a lot of studies being currently done right now, and a lot of assumptions and limitations. As I said before, the setting rate is unknown, so we do not know, how long the infected individual said the virus, whether it decrease or increase over time, and how it varies by different people. And also a lot of research gap in wastewater sample itself, like how weather, climate can impact the wastewater, the dilution factor, because, individuals, they flush wastewater with a lot of other stuff, and the decay and concentration method. So there are a lot of variability to successfully utilize wastewater-based EPI, but there are some advantages. So even though we cannot, back calculate the number of infected individuals, the wastewater testing can actually show the trends, whether it increasing or decreasing in the community. So the public health officials can take proper action or make the data actionable, implementing proper, lockdown controls if the cases are increasing. So we've been able to publish a few papers on SARS-CoV-2 RNA. First, we looked at the SARS-CoV-2 in wastewater in Louisiana, and then we published another article looking at how SARS-CoV-2 can implicate the current and future wastewater system management and operation. I showed you some data on the decay of SARS-CoV-2 in untreated wastewater. Showed you some data on how we used wastewater testing in high-risk area like university dorms, and were able to prevent a potential outbreak. And as I mentioned, the collection of wastewater itself, there are a lot of variability. So whether you collect grab samples versus composite samples. So grab sample is you just go at the treatment plant and collect it in one particular day or time. So that might not reflect the true the SARS-CoV-2 levels. Versus composite sample is, it's been collected over 24 hours and can actually use, composite samples for SARS-CoV-2 testing. So some of our future work that we plan to do is to evaluate the persistence of infectious SARS-CoV-2 in wastewater. So far, most groups are looking at nucleic acids, so viral RNA, to correlate with COVID-19 cases using as an early warning system. But we want to look at how infectious SARS-CoV-2, which are, viable virus, if it can be fecal oral transmitted throughout. So that had other implications in low-income countries where they might have higher chances of fecal oral transmission. So we want to validate the most appropriate and highly efficient concentration method. As I mentioned, there are tons of other methods and no uniform method right now. And what we are doing is also looking at new viral variants using wastewater testing. So there's a lot of other variants like UK variants, South African variants. So we want to determine the fate of SARS-CoV-2 and determine the circulation of SARS-CoV-2 variants. So and it's highly successful in high risk areas like dorms, prisons, and hospitals, where you can actually have a lot of controls. So the take home message is, we cannot actually back calculate the number of infected individuals. So there some challenge remaining to make the wastewater data actionable in terms of calculating the number of infected individuals. But it can help you kind of generate, the trends, whether it's going up or down, more in high risk areas. So we have a lot of successful stories using wastewater testing being a critical part of public health decision making. More than 200 groups working on this right now, and collaboration is key and communication. So we gotta collaborate with the public health officials and communicate using the advantages and disadvantages of wastewater testing. So it's the relationship between the wastewater treatment and public health protection. So another challenge is a lot of communities, they might not have centralized systems. So all the wastewater from the city, they might not end up in the wastewater treatment plant. So there might be some small or on-site septic systems or decentralized system where we do not know whether wastewater testing of SARS-CoV-2 can help mitigate COVID-19. So there are still some study gaps. So besides, SARS-CoV-2, we've been monitoring other enteric viruses and indicators in wastewater. This is just a graph showing you the concentration of these viruses in raw sewage in two different treatment plants. So as you can see right here, the first two graph here is the indicator cross phase and paper mild motile virus. So these are indicators, as you can see here, concentration about 7 to 8 log copies per liter in wastewater versus other pathogenic viruses, mostly enteric viruses like IHE virus, norovirus, which is the #1 gastrointestinal virus among all age groups. So we have genogroup 1, 2, and 4 norovirus, as you can see here, and enterovirus, adenovirus, which is a double-stranded DNA virus, polyomaviruses, and sapovirus. So what we are doing is looking at these viruses in the wastewater, raw sewage, and this is connected to the global climate change and water reuse. And today is the World Water Day, and with increasing population, we have to meet the demand of drinking water. So a lot of communities, cities across the world trying to reuse the treated wastewater for augmenting drinking water source. So we want to know how these viruses are inactivated or removed from wastewater treatment plant. So this kind of gives us an idea, how much concentration of these are in raw sewage, untreated wastewater. And then we can look at how it's being treated in effluent. So this is the study we conducted last year, looking at how the enteric viruses are removed in during wastewater treatment plant. So we collected, samples over like 18 months, and then we looked at the raw sewage before, going in the treatment. So that's the untreated wastewater, and it goes through, different treatment, primary, secondary treatment. Right here, primary sedimentation tank, activated sludge, and then we collected samples before chlorination, so before tertiary treatment, and after chlorination. So most of the typical conventional wastewater treatment plant, and after chlorination, it discharges into the river. So that's the treated effluent. And then what we were able to see was, cross phase and paper mile motile virus. They were actually kind of indicating the presence of the total pathogenic enteric viruses. So this is the correlation. So we were able to find highly correlation between cross phase, which is the newly identified DNA indicator of enteric viruses, and then paper mile motile virus and tobacco mosaic virus, which is a plant virus. So paper mile motile virus is also a plant virus. So overall in this study, we saw, norovirus genogroup 1, norovirus genogroup 2, human adenovirus. As you can see, log reduction of these viruses. So we saw 2 to 3 log reduction. And then cross phase was able to, kind of highly correlate. So the take home message of this is, the cross phase can be used as an ideal indicator for removal of enteric viruses during wastewater treatment. And we also conducted another study looking at, how reduction of human enteric viruses and indicator viruses vary during wastewater treatment plants. So we found norovirus genogroup 1, that's the most prevalent human enteric viruses. We looked at IgG virus, enterovirus, paper mile motile virus, and tobacco mosaic virus. So overall, we found high concentration of these viruses in wastewater that can implicate water reuse application. So that's the future right now. A lot of cities, even in India, a lot of cities trying to augment drinking water source with treated wastewater. So we need to know what's the fate of these viruses, including SARS-CoV-2 and other enteric viruses. So I left some time for questions and answers. So if you guys have any questions, I'm happy to answer right now.
Okay, thank you very much for your wonderful talk on actually the wastewater based epidemiology, current challenge and the limitations. Especially the focus talk was actually on the SARS COVID-19. I think we have actually certain queries in the questions for our speaker. I would like to put actually the first question I would like to actually put is the Dr. Muhammad Irshad. Doctor, he's asking actually, can there be any modification or the improvement so far the wastewater technology is concerned, so that we can take care of actually this SARS-CoV-19 virus infection is concerned or this spread is concerned? What's your take on, please, Dr. Samindra?
Well, there have been only few studies detecting the virus in treated effluent. So the wastewater treatment is able to inactivate the virus efficiently. Since this is envelope virus, it doesn't survive very long in the environment or even in the sewer pipes. So by the time it gets to the treatment plant, the infectious, the live virus might be already, inactivated. So what we are looking at is just the fragments of the nucleic acid like RNA that might be still detectable, but there is, I don't think, a concern with the fecal oral transmission, but the study needs to be done just to kind of, I guess, confidently say that this is not a fecal oral transmitted pathogen like other enteric viruses.
We have the other question from this Anjum Sabah. She's asking that, is it possible for SARS-CoV-2 to cross and contaminate other microorganisms present in the water bodies, making water more hazardous?
As I said, we do not know a lot because this is not a fecal, waterborne pathogen like other viruses. So the assumption here is, the virus that's being detected, it's not live virus, so it's not, infectious. So we are just looking at the viral fragments of RNA. So it might not be contaminating other water bodies, but the studies are very limited in terms of looking at it in surface water or river water or other, water bodies where there is more contact, recreational contact. So we do not know how hazardous, but with the knowledge we have, since this is an envelope virus and it doesn't survive very long, like compared to other enteric viruses, and it's a RNA virus, so it degrades pretty fast, So there is very limited, I guess, information about surviving this outside the host and having infection. So there is no data on that. So I don't think it cross-contaminate with other microbes and can make the water more hazardous. So there is no indication of that.
We have the one more participant, namely Mr. Rizwan Mudassir, is interested in asking, everyone is talking about the biodiversity conservation. Why isn't we are thinking about the conservation of a virus? So can you have a please comment on this observation from the
Uh,
participant?
well, it's a very interesting question. So I guess in terms of conservation of viruses, I think we want to conserve viruses that are, I guess, useful to us, mostly in terms of bacteriophages, viruses that kill, bacteria and eat bacteria, harmful bacteria. So I think but for viruses like, SARS-CoV-2 that's causing this pandemic, I don't think, people will like to conserve and most like to get rid of and eradicate this virus that, killed more than 2.5 million people within a year. So I think most conservation and of viruses are being done for bacteriophages because there are some advantages like with the phage therapy and the ocean phage, bacteriophage that can regulate the microbes in the ocean that can help the biogeochemical cycle. So for those viruses, I think um, it's good to have conservation, but I don't think for pathogenic viruses that can cause millions of lives. So it's difficult to kind of think like that way.
Okay, we have the one more, Sumaira Rashid. She is interested in asking in what kind of lab such SARS-related wastewater research was carried out. What were the precautions that were put in place for conducting such studies? She's about talking of the laboratory facility. Please.
Yes. So first of all, when we first went out to collect wastewater samples, we used PPE, so protectional equipment. So we used a suit that's used to collect the harmful pathogens in wastewater with mask and goggles and everything, gloves, everything like basic PPE. And once you bring in the lab the samples, you can actually pasteurize the water sample, which is just leaving it in high temperature. So the idea behind that is at high temperature, these viruses, if any of them are infectious, they can inactivate at high temperature. And then after that, we just follow with the concentration step and then extraction of nucleic acid and then PCR. So we need to have proper precautions. And I think the agency that monitors that had a very good regulation outlined, and everybody is following that in the US.
We have with Muhammad Sharjil Sofi, who is actually interested in asking, is there any study conducted on the presence of other variants of SARS-CoV-2 in the sewage?
Yeah, it's a upcoming study. So there are only few papers out right now, only two or three. They are used wastewater testing to monitor the new variants, but it can be done. So especially after now the vaccination is after vaccination, we need to evaluate how effectiveness is this vaccine, right? Whether it covers all the variants or not, and whether if there is going to be any other future outbreak. So in that scenario, wastewater testing going to be very beneficial because it can actually help you monitor the trends of COVID-19 and also look at the other variants that might be emerging in the community. And as I gave you an example of poliovirus, that's, we have still few countries that, still have poliovirus and they haven't eradicated. So using wastewater environmental surveillance, they can actually, get an early warning if there is any new variants, wild types, poliovirus circulating. Same thing with SARS-CoV-2. If any new variants circulating in the community, and using wastewater testing, we can identify, places where you need more vaccination. So if there is a community, where you detect a higher concentration of SARS-CoV-2, you can identify kind of location where or area where you might increase the number of vaccinations, so that way you can actually prevent some of the future outbreaks.
I think we have some, perhaps it is an anonymous attendee. He's asking or she's asking for, can viruses survive on the Mars?
Well, we do not know because there is no research being done on the survival of viruses on Mars. So I guess in the future we will find out, where more research is being done. Well, there's probably some life in Mars, so some bacteria, and wherever there is bacteria, you'll probably find bacteriophage, which is a virus that likes to eat bacteria. So they are collecting samples now, and when they analyze, they will find out what kind of bacteria going to be in those extremophiles environments and viruses that's associated with it.
Okay, I myself will be actually interested in Dr. Samindra regarding this. How would you actually foresee the technological advancements so far the STP technology are concerned? Apart from looking from the bacteriological or viral loads are concerned, because we have the technology and the advancements so far the treatment facilities are concerned. How do you see it in the future? What can be done so far in order to increase the efficiency of the sewage treatment plants, because they are hitting very hard so for the aquatic ecosystem health is concerned? What's your take on, please?
Uh, well, as I touched base on the climate change and the need for water, a lot of communities are treating wastewater and using it for drinking water. So the treatment plant itself, I think using advanced treatment with UV, ultraviolet light, and ozonation, and hydrogen peroxide with this advanced treatment, the water is safe to drink. The problem is the infrastructure, because water, it has to travel miles of pipes underneath, before it gets to your individual residential tap. So that's where the problem is, because even the water is clean and treated, by the time it gets to your home, the pipes, if they are not maintained or upgraded in certain time, you may have cross contamination with other sewage pipes or other contamination events. So the treatment plant, I think we have technology to make water clean from wastewater, and public perception is increasing, and they are understanding the concept of water reuse and using treated wastewater for drinking water. So the problem is the infrastructure of delivering tap water, not only in the US, but majority countries, we have miles of pipes underneath the ground that, deliver tap water in individual residential or businesses. So that's where the problem can or contamination can occur. So I think more investment in infrastructure and the pipe materials, I think that will decide the future.
Okay, we have this query from our faculty member, Dr. Rashid Jahangir. He's interested in asking at what temperature it is total dead or deactivated, since it is virulent in all seasons, and why it not is infecting the kids.
Well, in terms of temperature, the higher the temperature, the virus doesn't survive. So even in our studies, if the temperature is low, like at 4 degrees Celsius, like 10 degrees Celsius, it can survive longer. But if the virus is, at 70 degrees Celsius or, 65 degrees Celsius at higher temperature, the infectious virus, they will survive less. And in terms of infecting kids, I don't think there have been a lot of studies looking at kids because they have growing immune system, and not only SARS-CoV-2 but other influenza viruses not studied in the U.S. So I think there is a vaccine now, so not for children right now, but they are still doing more studies looking at the vaccination for children. But at higher temperature, most of the viruses are dead. The infectious viruses are. So that's why we do pasteurization at 72 degrees Celsius to make sure if there is any infectious viruses, they die before we proceed to the next step.
I think last thing this I would like to actually know from you, because the last couple of months you have responded to this COVID pandemic and have come with a good number of high impact factor journal publications. How this planning has come? Can you share with our researchers how it has come to respond to COVID-19 because of your publications?
Yeah, so my group was the first one to publish, the research in the U.S. There were a few preprints before us looking at this, wastewater testing in Netherlands and Australia, and we had a collaborative approach. So we communicated with some of our, researchers in those countries to kind of understand the process, and then we also wanted to work in low-income countries because, in high income countries like the US, Australia, Netherlands, we have centralized systems. So all the wastewater is collected and it's at the wastewater treatment plant. But in the low income countries like, certain parts of India and Nepal and, Bangladesh, a lot of these countries, certain part of Africa, sub-Saharan Africa, we have collaborators. We do not have centralized systems. So, a lot of wastewater is either discharged in the river without treatment. So you can actually have some contact, with contaminated surface water. So we are doing a lot of studies in these environments in decentralized system. But in the US, it was very easy because, the supplies, we were able to get it on time. A lot of countries, had trouble getting supplies because most supplies were directed towards clinical testing. So there were not a lot of research supplies for environmental surveillance or wastewater testing in the early months of the pandemic. But we were able to get the supplies on time, and we had, as I said, very dedicated students and researchers worked even during the pandemic to generate some data to publish. So they were very hardworking, and we were able to accomplish and we're able to secure funding and wrote publications. So it was a team effort. So not just me, but other individuals helping this research.
Okay, Let me thank Dr. Samindra for his wonderful talk and insights, especially on the COVID-19 pandemics and the virus load that we are looking to have actually in the wastewater treatments. It has given us some deep insights to how the future vis-a-vis the wastewater and the virus load will be actually in the years to come. I think I would again like to thank Dr. Semendra for his wonderful insights deep into this topic that was titled actually the wastewater-based epidemiology, current challenges and limitations. I once thank for extended discussions with the participants on this issue. So I would like to again thank with this, stay safe. Thank you very much for your wonderful talk.
Thank you so much for inviting me, Dr. Bhatt. Talk to you guys soon again.
Okay, thank you. I think we have another speaker with us. If he is actually able to hear me, Professor Dr. Larry Stevens. Can you hear me right now, Dr. Larry?
Yes, Dr. Bhatt, can you hear me all right?
Okay, you're welcome to this technical session. Let me introduce our second guest, that's the Larry Stevens. Just a second. He is going to deliver a talk actually on the spring ecosystems, a vital but threatened global water resources. As our worthy Vice Chancellor was actually also interested, I'm not sure whether he has been in a position to actually been able to log in, but it actually speaks of the importance of the springs that he has emphasized, because we have the thousands of springs actually. I'm sure the participants will get more deep insights, especially into what we call as the chronobiology or the spring ecology, as our yesterday's guest Marco Cantoni was also emphasizing that. Dr. Larry will be actually emphasizing more on the ecological components of these springs. So I will be giving a very brief introduction towards today's speaker. Larry Stevens is the curator of the ecology and conservation and director of the Spring Stewardship Institute at Museum of Northern Arizona in Flagstaff. To those positions, he brings more than 40 years of scientific research on aquatic ecosystem ecology, land and water management, advising many levels of governance, including tribes about those topics. After working as a biological technician and a whitewater river guide in Grand Canyon National Park, he began graduate studies at the University of Arizona, Tucson, and Northern Arizona University, where he received his PhD in zoology in 1989. Dr. Stevens served as ecologist for Grand Canyon National Park from 1989 to 1994, the editor of Red Lake books, and he has served on many ecological councils and committees. He is an evolutionary ecologist and a leading authority on the biology of the Grand Canyon region. In addition to his curatorial post, he also is senior ecologist for Grand Canyon Wildlands Council, a non-governmental organization, conservation organization, and is a conservation representative for the adaptive management of Glen Canyon Dam and the Colorado River through Grand Canyon. Dr. Stevens has worked on many scientific topics over his career, including biogeography, regulated river ecology, endangered species biology, focusing since 2000 on the ecology and stewardship of springs. He has authored or co-authored more than 115 scientific publications and several hundred reports. Dr. Stevens' recent publications include several books and numerous scientific and popular articles on topics ranging from rubber fly, taxonomy, water bug, biogeography, peregrine falcon foraging behavior, river and dam management, paleohydrology, spring ecosystem ecology, whitewater river running, and dragonfly ecology. The scientific interests relate to understanding biotic assemblage, responses to landscape development, and environmental change over time. He has supervised 4 BA senior theses, 29 master theses, 5 PhD dissertations, and 1 postdoctoral candidate. With these introductory remarks to his professional competency, I would like to request the Larry Stevens to share his presentation. Please, Dr. Larry.
Thank you, Dr. Bhatt. And please let me know if any of my communications are problematic, because we're many miles apart from your excellent symposium to here, my home in Flagstaff, Arizona. So please let me know if there's any problem. It's a pleasure to speak with you today. Dr. Bhatt has invited me to talk about springs and the ecology of springs. And this is a subject near and dear to my heart and the studies, the colleagues that I work with. We are discovering more and more about the fundamentals of springs and how they are small but very important ecosystems in the landscape, supporting not only a lot of biological biodiversity, but enormous amounts of cultural diversity, and the integrity of springs has become a major focal point for my studies. So I'll introduce you to springs in a lot of the world, actually. I will introduce you to some of the springs in our southwestern USA landscape here, but we're talking globally with this presentation. And so, why do we look at springs? Why are we interested in springs? These are systems where groundwater reaches the Earth's surface, usually flowing from the Earth's surface, but not always. Sometimes just sitting there. These are subsurface to surface linked, groundwater dependent, bottom-up ecosystems. In the background here is a deep seafloor vent, what I call a black smoker. These are fascinating ecosystems to look at with many dimensions to them. These might be the place on Earth where life originated several billion years ago. Springs on the seafloor recycle all of the Earth's water every 8 to 10 million years. Underwater, on land, everywhere, springs function as keystone socio-ecosystems, often hosting ancient life forms, many endangered species in the US, but also many rare and poorly understood, and many undescribed species as well. So these are gravely threatened ecosystems, as Dr. Cantanati probably pointed out in his talk yesterday. But they can also be quite resilient and sustainable and restorable if the aquifer that supports the spring remains intact. And so that's an important lesson for us. If we can maintain the groundwater supplies and the aquifers that support these springs, we can actually manage a great deal of biodiversity, a great deal of cultural diversity in a way that's sustainable. Springs might be our most sustainable ecosystems. So today I will talk to you about the distribution of springs globally and locally. We'll talk about the physical ecohydrology and classification of springs, their evolutionary ecosystem structure and function, and then how humans use springs, goods and services, and the threats that people pose to them. This is a first blush at a global springs map. We published this in a paper in 2020. What you notice most is that there are a lot of springs mapped in the United States, in the Mideast, in Southern Europe, and not much elsewhere in the world. This is not a complete map by any means. All the data on the distribution of springs in India, for example, or Brazil, are certainly not known to us. We would love to be able to add those data to this. And so if any of you listening have a good kind of documentation of the springs of India, we would love to hear about that, to be able to add that to this compendium. We guess there might be 50 million springs on Earth. That's Dr. Cantanati's estimate. Dr. Bhatt and others have indicated there might be 5 million springs in India alone, and most of those in the northern portion of the country. These are critical drinking water sources, as everyone realizes, with several of your states having extensive use of springs for drinking water and irrigation. And as I say, they're also vital components of biodiversity and ecosystems. Springs provide the base flow of pretty much every river on Earth that doesn't arise in an ice-dominated or a snow-dominated landscape. So pretty much all the temperate rivers on Earth are arising from springs, and springs provide the base flow, which is the critical minimum amount of water that, that those rivers receive. Few countries, India is moving towards a programmatic awareness of springs, but few countries in the world have that. Norway, and Australia beginning to have that, but not too many countries in the world have an understanding of what their springs are, where they are, what their status is ecologically and culturally. India has made some very great headway in beginning a national program of looking at springs, and so and therefore getting at the issue of how to better manage them, how to be better stewards of springs. This is only one map that I could find of springs, just the regions of hot springs in India, probably well known to many in your audience. Hot springs get a lot of attention. They're also among the most endangered kinds of springs because they're so often used for recreation, for resorts, for balneological or health purposes, and also for geothermal resources. So India is blessed with many hot springs, and that's great. But what is the status of these springs? How many of them are in sufficiently natural condition that we can actually understand how to manage them as natural and human facilitated ecosystems? Here in the United States, we have we map our springs on our U.S. Geological Survey topo maps. This is just a map of the named springs in the United States, but it shows some interesting patterns. We do not have, most springs are found in mountainous areas, the Appalachian Mountains and the Ozark Mountains in the eastern US, the intermountain western US, Sierra Nevadas, and in the Pacific Northwest, the Edwards Aquifer in Texas. So springs tend to show up in landscapes that are broken up, geologically broken up, where the edges of the aquifers are tilted and exposed, and that's where many springs arise. Flatlands, such as our North American Great Plains, are very low in springs density. In the case of Alaska, we don't have enough information on it. There may be a million or more springs in Alaska. Only 35 of them are named, however. So great areas of our country we don't know very much about. Here in my landscape, the Colorado River Basin, this is a river that provides water and power to about 40 million people. We've been doing a lot of work on springs here. The yellow dots are springs that have been reported, but not yet surveyed. The green dots are springs that we've been able to survey. So we're gathering information about it. This river basin, fairly large, almost 650,000 square kilometers. It's the dominant river of the Southwest. We've got 20,500 springs reported in the landscape. Probably many more than that exist, but because they're not, again, the mapping is so poor. But it's a really diverse landscape. Diverse geomorphology, chemistry ranging from very, very cold springs to geothermal springs. In this landscape, we have at least 330 species of springs-dependent taxa, plants, invertebrates, fish, amphibians that can only live in springs, and often only live in one or two springs. So a great number of endemic species. Coupled with that are literally thousands of species that we don't know enough about to understand their distribution or their habitat affinities, and some species new to science as well. There are 30 different cultures living in this landscape, all of whom place enormous value on their springs for religious purposes, as a clean water source, and as recreational sites. Interesting that this large river system, more than half of the flow of the river is derived from groundwater, from springs. That's a newly arising realization that that groundwater is supporting most of the population of the southwestern U.S. Now, in terms of trying to understand how springs work, I mentioned at the beginning that these are bottom-up ecosystems. The geologic context, the regional climate interactions affect the aquifer, and that feeds through to where the spring emerges, the geomorphology of the individual site, the microclimate associated with that, say it's a north-facing or a south-facing low or high elevation site. Processes like natural disturbance and productivity affect the emergence of that spring and result in a template of microhabitats and soils. Those microhabitats and soils are colonized by biogeographic processes, both active and passive. And so when one arrives at a spring and looks at it, one sees trophic structure and function. We see species interacting with each other. That whole system changes, of course, through time. And what you see 1 year at a spring or 1 day at a spring may be quite different than what you see at subsequent days, subsequent time periods. Humans step into this matrix and start to extract water and minerals and wood and wildlife and use the spring for various purposes and construct things in the springs. And those ecosystem goods and services and the human activities can affect pretty much all levels of spring organization. So humans can have a very, very profound impact on springs. This is a model that stands behind the database that we've developed called Springs Online. That's springsdata.org. It's available to anyone who's interested in archiving information about their springs and contributing that to better understanding of springs. Again, this is a diagram from a paper we put out on springs classification last year. All right, so where does the water come from? Much more than 99% of groundwater comes from meteoric sources. It comes from the atmosphere, and incoming precipitation as rain or snow infiltrates into the landscape. In arid regions, such as here in the arid Southwest and throughout arid regions of the world, more than 90% of the incoming precipitation is lost to evaporation. So only a tiny fragment of the water, a tiny portion of the water coming in from the atmosphere actually makes it into the ground. Snowpack, it turns out, is a critical resource for infiltration. As the snow melts, the ground is saturated, and that allows the meteoric water to sink into the ground. So springs in arid regions are particularly at risk because of reducing snowfall under climate change. And especially during droughts, very little infiltration happens. So, and some of our aquifers on Earth are incredibly ancient. In the Great Artesian Basin, some of the waters emerging from the ground there might be as much as 1 million years old. We have examples of springs here in just around me where the groundwater is 13,000 years old, meaning that during the Pleistocene times is when that water went into the ground, and now we're beginning to extract that at dangerously high rates. So the groundwater infiltrates and passes down through rock layers, hits what we call an aquitard, a layer of rock that is impervious to water flow, and the groundwater moves out. And so aquifers can be stacked. In our case here in the Southwest, many rock layers can serve as aquifers. They're stacked on top of each other. There is movement through those aquifers through faults in the landscape, and that means that there's progressive downward movement. And again, based on the tilt of the landscape, that groundwater may flow long distances. And as it flows, it begins in a cool and generally ion poor condition, high quality water for drinking. But as the flow path becomes longer and the water molecules move deeper, they generally warm and become chemically enriched. So deep aquifer springs are often not very palatable for humans. So the groundwater flow path is actually a very critical part of understanding how a spring is functioning. And excuse me. Around us, we have here in Arizona, we have springs that have very short flow paths, maybe only a day, not even a day, for water to infiltrate through fractures and faults in karstic terrain, move through the rock and emerge in springs. In contrast, deep aquifer springs, I'm showing a picture of Montezuma Well here in central Arizona. The groundwater flow path is 13,000 years. And so there's much transition of the water over those longer flow paths. And that makes it more difficult for life to inhabit those kinds of environments. As part of a springs classification effort, we put together a paper in 2020. This is Abe Springer, Ed Shank, and I, to describe the geomorphic types of springs. Most of our ecosystem classification, of course, is based on vegetation. That doesn't work very well for springs. The landforms that springs emerge from and sometimes create are much more diagnostic and much more provide much clearer information as to how to classify springs. And so we've identified about a dozen different types of springs, from marshy wet meadow helicrines, standing water exposure of springs, limnocrinic pools, mound forming springs, travertine forming springs, so recognized by the European Union, springs that emerge under pressure as fountains, hypocrenic springs where the groundwater actually doesn't reach the surface directly, but only through vegetation. And then lodic spring types, cave springs, hill slope springs, rheocrenic springs that emerge in channels, and then very dramatic geysers and gushets, waterfalls pouring out of cliffs. And here in the Southwest and in landscapes with stacked sedimentary sandstones, we get a spring type called hanging gardens. So an array of spring types all across the landscape. Some of these play differentially important roles in terms of what human activities they support, what kind of organisms they support, and what role they play in the overall landscape. Especially the pool forming springs, the limnocrines and mound forming springs, hanging gardens, and some hill slope springs may really promote the development of endemic species, for example. Here's one of the strangest spring types on Earth, I think. It's a mound forming spring, but the mound is actually ice. These form in high latitude settings, permafrost settings, and these can be more than half a kilometer or a quarter of a kilometer to a half a kilometer in diameter, 50 or 60 meters tall. The ice is building up as under pressure from groundwater that's rising. It actually uplifts above the surface. Because these then become not your normal permafrost settings, they become colonized by shrubs and even trees. So these are called pingos, and this is a spring type that we're rapidly losing as the Arctic warms, but very dramatic and strange spring type in Arctic settings. Now, as if the array of spring types wasn't complex enough, large springs, particularly, can host multiple different types of microhabitats, perhaps even a dozen different types of microhabitats. This is a splendid spring near us in Grand Canyon called Thunder River, and it hosts nearly a dozen different microhabitats. Each microhabitat is kind of performing in its own way, supporting its own species. The microhabitat may or may not interact very much with the rest of the spring's habitat, but it is one of the mechanisms whereby biodiversity can build up in springs. So each of these microhabitats supporting its own species contributes to the biodiversity in a very mosaic pattern. So let's look a little bit at the kinds of species that show up at springs. Plants often get a lot of attention, and what we see when we look at not only plants, but also invertebrates and vertebrates at springs, is that springs support not only wetland species, but also upland species. So it's not unusual to have coniferous trees or, clearly upland plant types showing up at springs. They're often facultatively opportunistic, and they might actually grow quite well in the periphery of the spring. And they influence the structure of the spring, as well as the animal life that might show up at it, and human activity as well. In addition to upland species, there are widespread wetland species. In our case here, Mimulus cardinalis. There are species that might be very widespread, but show up only at springs. Our helleborine orchid is one example of that. I'm sure there are examples of this in India as well. The array of non-vascular plants and algae at springs, Dr. Cantonati hopefully talked about some of his incredible research on the algae of the Alps at springs. Great biodiversity, many endemic species, enormous complexity to those assemblages. And some of those are widespread, some of them are very strictly springs endemic. And then we also have terrestrial plants that are found only at one or a few springs, highly localized endemics. So this is the array of species that show up at springs. And when you begin to look at a large number of springs, we find a really remarkable pattern that in one study, we looked at 243 springs in our region. Total area that we surveyed, springs are mostly small, so the total area surveyed was 10.2 hectares, quite a small land area, 0.0003% of the overall land area that we looked at. But the springs supported 620 species, which is about 25% of the regional flora. There's no other habitat you can go into and see 25% of the total regional flora here, almost 3,000 species of plants in such a small area. We see the same pattern elsewhere in North America, in arid lands and in mesic lands as well. So this is really remarkable. The biodiversity concentration at springs is enormous. Saving a few springs can save hundreds of species in a landscape. Overall in the US, only about 2% of our US flora are springs dependent, a rather small overall concentration there. However, there are many, many species that are springs dependent that we don't recognize yet, simply because the habitat descriptions have never been very adequate. There's a great deal of variation in the assemblages, of course, based on the springs type, water quality, whether the spring is shaded or not. Those all very much influence the flora at a spring. And in terms of kind of bad news about springs, is springs are places where we find a lot of weeds, and there is anomalously a positive relationship between the number of non-native plant species and native plant species. You would think that native plant species might create a situation that would resist the invasion of non-native plants, but that's not the case. So many interesting patterns here, a lifetime of work still to be done on the vegetation of springs in any landscape. Many opportunities, I'm sure, there in India as well. So plants are one arena of interest. Invertebrates are another enormous and very poorly explored realm of springs. All the species you're seeing here have some associated with springs as endemic species. Many of them we don't know very much about. Many of the worms, vast species complexes in some of the amphipods, poor understanding of dryopoid beetles, of flatworms and ostracods, all of which have many endemic populations at springs. The poster child, the most diagnostic group that is represented here are the spring snails. These are the Truncateloidean snails. They're small, very small species, just a couple of millimeters in size, but distributed globally. I'm sure there are many in India. It's often the case that there are one or two, that a species of spring snail might only live at one or two or three springs. We've got almost 200 species in this group identified in the US as so far, and more species being described each year. So enormous biodiversity of these. They live in New Caledonia, in Australia, in Africa, in Europe, everywhere in the world, except Antarctica. So spring snails are a real poster child, if you will, for endemism at springs. Enormous biodiversity, and we're still just beginning to plumb those depths. I had a thought in this because of your incredible emphasis on human health, on water resources, and on parasitism. I began to think about springs as possible landscapes that either protect or promote strange, complex life histories. In the lower left here is a picture of a succineid snail, Oxyloma haydeni, that is subject to a Leucochloridium trematode parasite. The parasite occupies the snail, and as it matures, it produces a sporocyst that flashes pink and green very brightly. The parasite drives the snail to the edge of the habitat where it's attacked by a bird, thinking that the flashing sporocyst is a worm. The parasite completes its life history in the intestines of the bird, and the bird then poops out the eggs that the snail picks up again. So this is happening at only 3 springs in Arizona, and it's a snail that is found at Vasey's Paradise and 2 other sites. the only places in the state of Arizona. And oddly, the snail has host shifted from it occupying a niche where it was feeding on dead and decaying monkey flower, and has shifted onto non-native watercress in its habitat. So many, many interesting questions about the life history of the snail. The Leucochloridium parasite is just absolutely fascinating. It matures in August when the snail matures. And the bad news for the snail, of course, is the parasite doesn't actually kill the snail. The parasite can produce several generations of sporocysts, by which time the snail is completely blind, completely under the control of the parasite. So this complicated life cycle, you know, it's something that pretty much has to only can only evolve in a very, very predictable environment. So it may be that springs are a place where such associations can develop and then expand out from them. That's as far as I can tell, that's an unexplored topic of research, but there may well be something to look at there. Now, in terms of the vertebrates that live at springs, as far as I can tell, there's only one bird group that is really a spring specialist, and these are some of the cichlid dippers, which nest behind spring-fed waterfalls in arid regions. But many other species, some widespread, like our bighorn sheep here in the Southwest. If the only water source is springs, then they become a springs-dependent species. Several species of snakes, quite a few fish. The famous case of the pupfish in North America, a big radiation of endemic fish, pretty much all of which are spring specialists. Quite a few amphibians also in this list, and a few voles and other mammals that are spring specialists. So not as many vertebrates are spring specialists because the habitats are small, but some very interesting life histories associated with springs in terms of the vertebrates. Springs also host a large number of non-native species, as I mentioned with plants, but also the red crayfish is becoming a global pest at springs, a non-native species that's being introduced all around the world, either accidentally or in some cases intentionally. It's widespread in Europe, all throughout non-native parts of its range in the New World. Bullfrogs, several non-native mollusks, many non-native fish introduced into springs. Because springs are such small, tightly evolved ecosystems, introducing these kind of predators into those systems is devastating to the springs. And so a lot of our work in terms of preserving springs has to do with controlling non-native species in them. So that's about the organisms that live at springs. Let's look at springs as ecosystems now. And the first place we go is to Silver Springs in Florida. This is a large spring pool system, limnicranic spring pool system that actually has enough flow to create a river. This is perhaps the largest spring in the United States, about 14 cubic meters per second. It's also the most extensively studied springs ecosystem in the world. Eugene Odum in 1957 produced the first analysis of ecosystem ecology by studying this spring. And it's had more than 100 peer-reviewed ecosystem studies conducted at it so far. So it's a very well-studied spring, got a big array of predators, including American alligators, very predaceous fish, an array of invertebrates. In these mesic environments, not so many endemic species. That's kind of interesting, at least as far as we can tell yet. Although, in all likelihood, there are probably many unique diatoms and perhaps microcrustaceans in these systems. An analogous, well-studied limnocrine mound-forming pool is in central Arizona. This is Montezuma Well. Dean Blinn and his students studied this in great detail. There may be a couple dozen publications on this springs ecosystem. The rabbit in the system is an amphipod, Hyalella montezuma, endemic to the spring. During the daylight hours, it has to race away from the shoreline vegetation because there are visually feeding invertebrate predators that are attacking it. So the amphipods race out to the middle of the pool. They're a little bit sensitive to light, so they sink down a few meters. But as dusk comes on, up from the ooze on this 17-meter deep pool come 4 species of leeches that can detect the clicking legs of the amphipod. So the amphipod population then races back to the shoreline vegetation where they cling motionless to the shoreline to prevent being detected by leeches. So they spend their whole lives racing away from predators during the day and again during the night, have to live all their life cycle and breed and reproduce in the middle of that. They're also subject to parasites in this system. So this is a warm, fishless system, very much enriched by calcium carbonate, but also has a kind of a kicker of arsenic that may play a role in the endemism here. One time in evolutionary history, over the last perhaps 100,000 years, a female water scorpion, this is actually a hemipterous insect, a gravid female, flew into the pool from the outlying landscape and didn't die from arsenic poisoning. And so her offspring created a new species. So the water scorpion is endemic. The world's smallest giant water bug is there. One of the leeches is a new genus to science. Montezuma Well has the highest point source of endemic species of any point that we know of in North America. So it's again, the environmental harshness, the constancy are very essential in the evolution of endemism here. Fascinating spring, very well studied. Now, in looking at springs from an ecosystem context, again, the dimensionality, multidimensionality is just pretty fantastic. Springs are highly productive, can be highly productive systems. Odum found levels of primary productivity of up to 6 kilograms per square meter per year in Silver Springs. That's the highest productivity I think I've heard of on Earth. That internal productivity is facilitated by clear water, by sunlight, and that productivity radiates both laterally out from the spring, perhaps down into the groundwater and through the spring brook downstream. The spring is also subject to kind of lateral import of organic material, of leaf litter, of animal life being attracted from the surrounding landscape to come into the spring, and animal life within the spring, of course, radiates out from the spring. So this subsidy exchange is enormously complicated and has not been well studied. Great opportunity for PhD level work if any of you are students and interested in really high level ecological work. Also, nutrient spiraling in a spring, very tight nutrient spiraling within the body of the spring, but springs they're a bit ecotonal in their structure. In other words, there's transitions as you move through, and the nutrient spiraling increases in length as you move from the spring into the spring brook. So as a multi-directional subsidy exchanging system, springs are just absolutely fascinating ecosystems. Humans have used springs all throughout our evolutionary history. Cuthbert and Ashley, 2014, found they reconstructed the paleohydrology of Olduvai Gorge in Africa and found that humans have been associated with springs there for more than 2 million years, and our ancestors there for more than 2 million years. That association with springs may have more complexity to it than we initially think. Yes, humans would have retreated to springs in times of drought, but springs are notoriously ambush sites, not only for humans attacking prey, but also for large carnivores attacking anything coming into the spring. Early humans probably faced many situations in which large predators were waiting for them at springs, and that may be part of the reason we find their remains at these paleo springs. Here in North America, the whole southern portion of North America, about 1,000 years ago, was affected by a belief system called the horned serpent. Has many names in many different cultures, been described by a couple different authors, but this was a creature that brought water to the surface of the earth, a divine being, and has many attributes, including impregnating bathing women at springs. So many of our tribes don't allow women to bathe at springs. But it shows up not only in petroglyphs, but also in pottery all through northern Mexico and the southern US. So humans have a long history with springs. And I'm sure in India there is a wonderful history to be told, and I would love to be able to talk with any of you about that history. In Israel, the Siloam Tunnel has a long history, a couple thousand years of water being delivered from a spring to the interior of Jerusalem. Here in North America, people build houses on top of springs to provide cooling environments for apples and wine and etc. So long history of use of springs for many different human purposes. Here, very few examples of economic analyses of springs. One such study in the early 2000s found that 4 springs in the state of Florida brought in $60 million per year to the local economies, and that they attracted 2 million visitors per year. So in some cases, the recreational visitation of clear water, cool water is great. What's the global economy around visitation to hot springs? Many billions of dollars. In addition, we've been learning more about what the U.S. drinks, and through Peter Gleick's work, and Americans drink more than 50 million bottles of water per day, many of them labeled as springs water. That's probably a $15 to $20 billion industry just in the US. The overall industry around the world is probably closer to $50 billion per year. So the economics of springs, again, not very well studied, but really quite remarkable in terms of the economic values, not to mention all the communities around the world. The city of Vienna, all of its water supplies come from springs through an aqueduct system. So many interests in the economics of springs, but not very well studied. Humans do a lot of things to springs, and this section is what humans do and how to evaluate that. Groundwater pumping for agriculture, for potable water supplies, for livestock watering, all can affect the geomorphology of the spring. Some of those uses can pollute the springs, pollute the groundwater, and therefore the springs that are emerging from those groundwaters, breaking up the habitat, diverting the flow, recreational impacts, as we just talked about, non-native species invasions into springs, and of course, climate change. All of these are major impacts on springs around the world, but we haven't really understood how much, the extent of those, or how these different kinds of impacts affect. I've engaged in a study right now with Dr. Bhatt and about 55 other co-authors. We've gathered information about the springs in the regions shown with red dots here. Each of these landscapes or countries has done some work on their springs, and we've pulled together narratives on those landscapes. So we're gathering information on springs. Right now, the study includes approximately 250,000 springs around the world. Obviously, there are big portions of the world that we don't have much information on yet. Russia and China have not encountered the literature yet. Many countries in South America and Africa, we've not encountered direct literature on yet, and Southeast Asia as well. And not much information in Antarctica, but gradually trying to pull this information together and present it to the world. For the 58 co-authors on the paper so far, talking about springs in their region, this is the frequency of times that different disturbances have been mentioned in their work. As you can see, more than half of the springs are affected by water supplies development, either for potable sources or other purposes, for agriculture, especially livestock propagation and watering, and also mining. And here I'm including with mining aquifer depletion for some of these water supplies development. So more than half of the springs that we've received reports on are being affected by these 3 topics. In addition, visitation, geomorphic alteration is probably going on with many of these springs, and then groundwater pollution are all big deals. We find more concern about climate change going on in lower latitudes and lower elevations than in higher latitudes and upper elevations. Climate change effects include this issue of reduced infiltration and changing habitat structure. So climate change is an emerging issue and likely to get much more significant over time. But this is the first look at what the factors are that are threatening the springs of the world. So it's not uncommon to find a graph like this, the hydrograph over time of a large spring dropping in, declining as groundwater depletion goes on. This is a spring in the Verde Valley of northern Arizona, and it's one of many examples of this kind of reduction in flow pattern over time. When this occurs, the structure, the ecological structure and function of the spring changes from there's some normal variation in flow that is able to maintain vegetation life. But when groundwater is pumped, that typical pattern of variation in flow drops, and the spring begins to dry out. Groundwater can be completely depleted, completely transitioning the spring from an aquatic system to an upland system. An example of hydrarch succession that's going on at our springs through human activities. Livestock watering all around the world is a major issue. This is a relatively easy problem to manage because one can fence the spring and prevent the livestock from getting into the water, provide water to the livestock just off site, and allow the spring to function. The sources of springs are where the biological intrigue, the biodiversity generally occurs. So if you can keep livestock away from that source, and you can do so quite safely, and actually probably better for the livestock as well, to keep them from wandering into these somewhat treacherous landscapes to water. In North America, we also have, in the US anyway, we have a legislation that protects wild horses. Wild horses do a huge amount of damage to our springs, but they are protected under the Wild Horse and Burro Protection Act, and therefore they can't be managed by our federal agencies. So there are clear policy limitations to what we can do to protect our springs, and some of them are quite unfortunate. Everybody loves horses, but again, fence the source, provide horses with sufficient water off-site, and don't allow the populations to grow to the point where they drink up all the water and then die of thirst. That's a terrible fate for anything. Climate change, we have basically three things we can do in relation to climate change. We can avoid it by moving away. We can figure out how to tolerate it, not very satisfactory, or we can try to adapt to it. Our adaptation needs to be smart. We need to improve the research, basic understanding, apply that understanding to planning, implementation, and use that information and implementation to feedback and improve our management of springs. So the winds of change are blowing very clearly, and it's time to figure out our adaptation strategies for protecting groundwater and the springs they support. It's very common to see maps like this showing the predicted climate changes over the next 50 years or more in landscapes, with climate change happening in a rather patchy fashion across most continents. Here in the Southwest, much concern about low elevation climate change, less going on at the upper elevations, but that's not matched by what's going on at northerly latitudes, where climate change is really rapidly affecting all manner of ecological function and human access to water. So these are very dire warnings about the future that we must take steps to work on. Here in the US and elsewhere in Australia, in Europe as well, wildfire frequency is increasing. And this is a manifestation of climate change. The frequency and intensity of wildfires is exceeding the ability of our forests to regenerate. Another wonderful topic for somebody looking for a good PhD project might be the question of, do springs function as regeneration foci after fire? Springs are wet patches in the landscape that may undergo a reduced fire intensity, and therefore allow populations of plants and animals to recover following fire. So some fascinating research still to be done there. So I'd like to summarize this portion of the talk about the global demise of springs ecosystems by asking who's to blame. Who's really to blame here? And I identify 5 issues: homography, policy, the lack of interdisciplinarity in our education systems, the limited representation, and an underinformed public. So I'll tackle these one at a time, just briefly. If you Google on springs, what comes up? Gymnastics classes, sometimes computer programs, auto parts stores, the season of springtime, and then strange gatherings of hundreds of thousands of unmasked springtime students, especially in the U.S., also in Mexico as well, and elsewhere. So the definition of a spring as a hydrologic function doesn't even show up in our dictionary until the 3rd, 4th, 5th definition. English may not be the best language for springs research. As I understand, the word sota in Hindi is spring, but Italian has the words sorgente, manantial in Spanish, Juan in Mandarin. All these are very specific words for springs, and those might be more useful than our word for spring, which has been preempted by all these other uses. So that's the issue of homography. Basic language issues may be partly to blame for why we're not paying enough attention to springs. Policy has also been quite a problem. Here in the US, we define wetlands as being surface water-fed systems through some publications early on. Springs, which are obviously wetlands, are not regarded as wetlands in the United States. This is a big deal. So our policy has excluded springs from being considered in the Clean Water Act and our other policies. This is also a problem in Europe and in Australia, where there seems to be an overemphasis on the surface water component of wetlands, not appreciating springs as being these headwater wetland ecosystems. The lack of policy and the failure to acknowledge the link between groundwater and surface water means that springs fall between the cracks in legal protection. This precipitates a lack of policy and guidance about springs in general, lack of funding for springs ecohydrology, lack of research on springs, and has produced a lexicon, a language about springs that's deeply confounded by bureaucracy, and resulting in our failed groundwater and surface water stewardship. Groundwater management in the United States is in shambles, and it's quite unfortunate because so much of our water supply is actually based on groundwater, and it's very short-sighted. Groundwater, this has led to groundwater pumping and pollution and flow diversion and livestock management without any constraints. This also has led to the failure to guide and support research and stewardship of springs and paying basic attention to them. And a manifestation of that is here are an array of books published in the last 20 years that virtually do not mention springs. Wetlands by Dr. Mitch and Gosselink is basically the Bible of how to manage wetlands. Only about 3 pages in that book deal with springs, but enormous kind of efforts: ecology of freshwater, drought and aquatic ecosystems, assessment of conservation values of freshwaters. None of these books actually mention too much about springs. We're trying to rectify that. We have a few recent books that address the issue of springs, but this is kind of late in the game, and we need much more attention to the integrity of springs and the groundwaters that they represent. The third failure, I think, is that springs are, even though they're quite small, they're incredibly complicated. It may be too multidisciplinary a field for most scientists to tackle them. This is the array of sciences one really needs to be familiar with to understand the ecohydrology of springs, ecohydrogeology of springs, if you will, and to be able to effectively restore and monitor and study them. This is more disciplines than most people get to look at ever. And it takes a real kind of generalist perspective to be able to get through the basic literature on these fields to be able to begin to look at their role in the ecohydrogeology of springs. There's also an issue of the failure of art to depict springs. These are three sets of movies in the United States that deal with springs. Actually, Bannons of the Spring is a French film. I strongly recommend it. The second session of it is the one that is most informative. 10 versions of The Creature from the Black Lagoon were filmed at Silver Springs in Florida, where Eugene Odom did his work. He didn't encounter the Creature from the Black Lagoon while he was doing a study, but nonetheless, it's a great place to shoot movies. Our Franklin Delano Roosevelt recovered from polio partially recovered from polio by going to Warm Springs, and there's a wonderful movie about his return to politics and the role he played in saving America from many threats. But relatively few movies deal with springs. And springs are rarely depicted in art. Again, there's some recent projects, some really, really wonderful art by Rosalind Driscoll, for example, taking up this topic of springs and beginning to pose, I think, fairly profound questions, visual, artistic questions, and cultural questions about springs by doing her art. So there is some forward movement here, but it's a long legacy of failure to appreciate them. All this results in underinformed and at least in the US and probably elsewhere in the world, a very polarized public. Humans tend to guard their resources quite jealously, and for all the impacts that we're having on springs, many springs that are under private management are off limits to even well-intended and scientists that would not divulge information that would harm anyone. So this polarization is really quite an enormous problem in the United States. I hope it's not the same way in India, but it really poses a great challenge to the management of springs. So we've seen this poor language, poor policy. Can't emphasize the inadequacy of the mapping data enough. There's a poor understanding of biotic and cultural significance of springs. The dominant white culture in the United States is, I think, one of the few cultures in the world that doesn't regard springs as being sacred places. We see springs as our culture sees springs as a commodity. Every other culture in our nation and most of those around the world, I think, see springs as having this deep, profound spiritual significance. We have very incomplete science. We know a little bit about a couple different kinds of springs, limnocrine pools, hot springs, but not much about the other kinds of springs. Great polarization in the part of the public, greed and indifference to the issues, and very much uninformed management. So the solutions here that Marco and some of our other colleagues have proposed is improving education, improving the mapping, improving information management, resolving classification, informing public policy discussion and ecological reconciliation to bring springs conducting inventories, studying springs-dependent species to get a better understanding of what they are, better understanding of the sociological condition of springs as well, the role of springs in landscapes, including their economics, and improving springs, spring type-based restoration options. We don't really have the technology understood yet for restoring different kinds of springs. To that end, myself, Jerry Ledbetter, and several other colleagues Abe Springer have started an organization we call the Spring Stewardship Institute to provide online free tools, trainings, and advisement for people who are managing springs. And these tools, I mentioned Springs Online at springsdata.org is a way to archive, inventory, assess, monitor, report, and study springs ecosystems. So if you're interested in finding out more about that, please get a hold of us. This is an aquatic moth, a cramped moth in the genus Petrophila. We just discovered a couple more species in our local landscape here. It's the larvae are fully aquatic. The adults stay right near the springs to feed and reproduce. So the Springs Institute, Spring Stewardship Institute provides these secure, easy to use relational information management tools that are designed to improve understanding, discussion, and stewardship of springs around the world. Right now, we've mostly focused in North America. We've got about 160,000 springs in North America in the database, 1,100 users, including many agencies, tribes, nonprofit organizations, researchers, and the public. And we do trainings and workshops everywhere. We've offered to do a training in India, perhaps over Zoom, or perhaps as the COVID winds down here and there in person. We'd be happy to do so, because India is making great strides in recognizing springs as important features. And we laud the Indian country for taking up this banner. It's critically important that we understand how to protect our groundwaters, and the springs that are often our best evidence of that. In Springs Online, it's a relational database, so it allows you to ask all kinds of questions that have not yet been addressed. Water quality relations to aquatic and riparian fauna, the role of the solar radiation budget in relation to elevation, climate, bedrock geology, et cetera, and their influence on the biota and on the human uses. Having a relational database like this, a very powerful tool, very easy to use, and quite enormous in its capacity to assist with the task of better improving understanding of springs. And from these kind of efforts, we can begin to actually get students out there and get willing volunteers out there to help do restoration, opening up habitat, trimming back non-native weeds and whatnot, reintroducing missing species. So there are many examples of this going on, some great examples in our landscape and elsewhere in the world. And we hope that this idea catches fire and people take on the issue of better management of springs. These are small but critically important features for human well-being, for nature, and really they provide a true test of the human spirit in terms of protecting and conserving them and recognizing them for the importance that they have. This is a different kind of conservation topic. Protecting and managing small points in the landscape that are highly influential in those landscapes is the conservation theme here. So I hope that this talk has given you some insight into springs, and be happy to entertain any questions. Dr. Bhatt, I'll turn it back over to you. I can't see the chat box here if there are any questions.
Thank you, Dr. Larry, for your comprehensive and in-depth talk on the springs, where you have touched on the geology, geomorphology, economics, as well as the art. I think it has opened the windows for a critical thinking on how we should look for the springs as a resource, not from the hydrogeological point of view, but from the ecosystem services. I think it has opened the window for a discussion also for our participants. There are certain queries and the question is vis-a-vis your talk. I would like to actually communicate you. The first question is from Rinchen Angmu. He's interested in, he wanted to know, does digging bore wells reduce or dries up springs or affect the natural springs? He's actually from the Ladakh, that's a desert. Would you like to comment on this, please, Dr. Larry?
Could you repeat the question one more time? I'm having a little bit of trouble hearing.
Yes, he is interested in know, does digging bore wells reduce or dries up springs or affect the natural springs? Digging he's talking of.
If one is hand digging wells, usually there's not much of an impact on the aquifer. It's when we come to mechanical bore excavations that go deep into the aquifers. In those situations, they can definitely tap the aquifer out, and it generates a cone of depression that can extend for some distance. It's quite important to understand where the water is actually coming from and how old the water is. Because if there's slow recharge, then extraction of deeper aquifer waters may take a very, very long time to recharge, if that makes sense.
I think we have another comment and the question that pingos can be found in the Tibetan plateau, especially in the region which is around 4,500 million meters above sea level. If yes, how can we differentiate it from the outside without digging? If no, why?
That question may take some beer to answer. Humans are extracting water at ferocious rates from many aquifers. I think it's important to look at the societal need for the water. How much do we need to take, and how well can the aquifer respond to it? One thing, again, by doing a bore test, you can look at the cone of depression and see how quickly it recharges, and that should help answer the question. If the recharge is very slow, then extracting too much water will be extremely detrimental to that aquifer and to the springs it supports, and ultimately to the society that's extracting the water.
We have another question from the Palak, who is interested in to which extent the dwindling springs has affected the parasitic diversity, because you are talking about parasitic diversity. Please throw some light, sir, on it if they are interested in that. Parasitic diversity.
So I just began thinking about this question because of your invitation for me to speak at the symposium. How do complex life cycles evolve? They must require very stable, predictable ecosystems. So liver flukes and many of the trematodes have notoriously complex life cycles, and I have not read much about how what the conditions under which those life cycles might evolve, but it could well be that that evolution can be facilitated by springs. I don't know that for a fact. Certainly, we only see. I guess there's probably not been enough research on the parasites that we encounter at springs versus, say, in streams or in ponds and lakes. So I think there may be a fruitful avenue for research in that whole topic.
We have another comment that's from Aditi Mishra. Which type of invertebrates are mostly found in springs?
Which type of which are found mostly in springs?
Invertebrates. Which type of invertebrates?
Sorry, invasive species?
Invertebrates.
Oh, invertebrates. Ah, good. Well, again, we're limited by the number of studies of springs around the world. The invertebrates that we see commonly at our springs in North America, in South America, and in Europe are some species that actually come out of groundwater. So groundwater dependent species like isopods and other creatures that may occasionally flush out into the spring. And we have examples of blind cavefish and blind crayfish, sometimes other organisms that actually emerge at the spring, but those are actually species that live in the groundwater itself and are only perhaps flushed out occasionally, so available. We discovered a bioluminescent isopod at some springs in southern Texas a couple of years ago. And this is quite shocking because isopods are not typically bioluminescent. It hasn't been described yet, but that's probably an example of an aquifer-dependent species that gets occasionally flushed out. Then the common aquatic invertebrates that we encounter at springs that tend to be endemic are several families of aquatic beetles, a large number of aquatic mollusks, a fairly large number of amphipods, and microcrustaceans, ostracods, etc. And so it's an array of arthropods, but also I must mention aquatic mites. Although we have only described about 5% of the aquatic mite species on Earth, there are probably many species still to be discovered. So arthropods in particular, but crustaceans as well as insects, the annelids, the flatworms are also rather poorly known taxonomically. There are not very many people who study the taxonomy of annelids, for example, in freshwater settings. So again, much to be learned there, but quite an array of species. In our southwestern landscape, we've now recognized 1,500 species that are springs dependent. I mentioned 330 that are in the Colorado River Basin, but across the southwestern portion of North America, at least 1,500 species of cranobionts or springs dependent taxa. Mostly those are invertebrates, although some fish and a few reptiles and amphibians.
So Mr. Adil Hamid, one of the participants, is interested to know, does the human-based conservation and management of springs harm the ecology and environment? If so, then what are the effective ways to protect these threatened ecosystems?
Very good question. I mentioned the simple practice of fencing a spring to keep livestock out of it. That alone, if we could solve that problem, we would save many ecosystems and species with that simple practice. It does mean managing springs in an intentional way, but humans have, interesting thing about springs is that humans have, as I mentioned, have pretty much always been at springs. There's no such thing as a large spring anywhere in the occupied world that has not had human imprint on it for thousands of years, or tens of thousands of years, or perhaps in Africa, millions of years. So humans have always been associated with springs, and the real question is, how can we manage our behaviors and our desires for the resources of springs in a way that is maintains the ecological integrity of the spring, and therefore the sustainability of it, and ultimately, because these are expressions of the groundwater supplies that we so rely upon here as humans, that ultimately it's to our own well-being to manage springs appropriately. Simple practices. If a spring is up on a hill slope and the water is being managed, of course, at the source, constructing a stepping stone trail so you're not eroding the hillside. Very simple measures, very inexpensive and easy to accomplish, can be very effective at conserving springs.
Mr. Ashif Hussain has a query regarding how can we measure the ecosystem services provided by springs, particularly groundwater recharge.
Yeah, intentional recharge is a new and developing field. In arid regions, excess water can be directed to faults and fractures that are fast paths for infiltration, and that takes a fairly sophisticated geologic understanding of the landscape and the stratigraphy. But it's being used in Australia, in several other portions of the arid portions of the world, and it's a very provocative and interesting kind of topic to take up. To redirect the excess surface water back into the ground makes perfect sense if we can figure out the geology to be able to do that. Putting catchments on those fault systems so the water passes quickly into the groundwater is quite essential for those to work.
One more comment from the Funsuk is, can aquatic organisms present in the springs can act as a biomonitoring species of the particular habitat? Can you please explain it with suitable example? Biomonitoring.
Yeah. Okay. Again, I'm having a little bit of trouble hearing. One more time with that question.
Yes. Can aquatic organisms present in the springs can act as biomonitoring species of the particular habitat? Can you please explain it with example?
So if the question is how to do biological monitoring of springs?
Yes, and they are interested in actually aquatic organisms can act as a biomonitoring tool. So for the spring ecosystems are concerned.
So if I understand the question, biomonitoring of springs for specific aquatic organisms, it varies by taxon. So for say fish or some some aquatic organisms, snorkeling might be one way to accomplish biomonitoring. For fish in particular, electroshocking if the water is not too highly charged with calcium carbonate. In some cases, the biological monitoring mark recapture methods are appropriate. So we have many examples of aquatic insects that can be monitored by, putting a number on the carapace of the insect, and then seeing and then resampling to see if those are recaptured from that, developing a population estimate over time. Does that help answer that question?
I believe so. I think before we actually close, I have certain these questions for the Larry, because I am actually a little bit interested in the spring ecology. Regarding this, so for the journals that are actually published, like we talk of rivers, we talk of wetlands, we talk of reservoirs, we do have a journal at the international level. What has happened wrong that we do not have come up with the journal that is specifically targeting the springs? Do you have
Yes.
some quick comments on that, Dr. Larry, please?
I've thought many times about taking up the editorial duties associated with that, and it would interfere with my research. Because I have been a book publisher and served as an editor on several different journals. It takes up a very large amount of time. There has not been support for it. Were there support for it, I think we could easily put together a global editorial board, and there is so much interesting research going on about this, about springs. We need to talk Marco into it, Marco Cantonati into it, and several of his colleagues, all of them very productive writers, and it's a big commitment to start a journal. And right now, some of the projects that we're working on are pretty large in scope, and so far other journals have been able to handle it. But I completely agree, we need a journal about springs, and springs ecosystems in particular, and their management, stewardship, restoration capacity, and their socioeconomics, etc. So a good idea. Let's talk about it some more.
Okay. I think if I have heard correctly, you are just in your talk referring to the Florida Springs, that much of the biodiversity has not been actually reported in the Florida Springs, although we have a good number of springs from the Florida itself. What could be the reason for that, that we have not been able to actually see the reports reporting the biodiversity within the Florida Springs?
Yeah. In the case of the spring snails that I talked about, one individual spent his whole life dissecting the reproductive tracts of snails that were only 2 millimeters long. It took his entire life for him to describe almost 200 species of spring snails. The techniques have to include, of course, morphological description as well as genetics. So it becomes very sophisticated and very taxon specific. Therefore, we need more people in taxonomy, for one thing. That seems to be a dying art. So many of the groups that I work on, the individuals who are experts are aging, or perhaps they've already passed on, and it's very difficult to get to even understand whether or not the species you've got is a new species for science or not, in many cases. And not that many people seem to be interested in freshwater worms, for example, or and where we're going to find another individual willing to devote his or her life to the dissection of reproductive tracts of spring snails, I don't know. 20 new species were just described in New Caledonia, for example, a long way away from the Gondwana land that it once was related to, and probably from where those spring snails originally originated. So I think boosting taxonomic studies is one avenue. Finding people that are interested in these taxa and engaging them more in springs related work would be helpful. But I'm not kidding. There are thousands, tens of thousands of species not yet described from springs that are awaiting description, and oftentimes we only discover them through some genetic analysis. eDNA is providing some insight into that, but still those tools are in the early stages of development, and much more to learn and apply there. It's a really wonderful field. Yeah.
Yes. To what extent you are convinced that the springs have not been found in the right places so for the conservation biology and the international context is concerned? as you rightly said, so for the policy makings, if when we talk of the Indian context, we could not find the separate springs. So for even the water policy context is concerned, that is actually 2012 is concerned. So what has happened wrong with this? So for these springs are concerned in the arena of conservation biology in the global context.
Well, I wish that policymakers would listen to scientists. That's one wish that I would have. It's usually not the case. And unfortunately, what that means is it will take a crisis to really elicit enough response. And by then it might be too late. In many cases, it may be too late. Tapping a Pleistocene aquifer for a water supply is being done in North Africa, in the central plains of the United States and Canada, and elsewhere. Once we lose those, once we deplete those water sources, there is no more water. And enormous canal systems take years, decades, sometimes centuries to build, and may not rescue us in time. The issue is incredibly important, and politicians are incredibly short-sighted. And trying to get the appropriate policies for long-term sustainability of groundwaters and the springs they support is the leading challenge, I think, in terms of water management for the world.
Okay, I think it may not be in a position to take more questions from the participants right now, because we have already had an extended discussion. A lot of questions have come actually on the springs. I think it is a time to actually thank you to this Dr. Larry Stebbins for his thoughtful lecture and the talk on the spring ecosystems. Again, a big thank you to Larry Stebbins. And I will actually make an announcement for this audience regarding that there will be a short break of 1 hour. We will be back at 12 o'clock again, when we will have Dr. Sarah Navrin, that she will be delivering a lecture on that. Till that, it is goodbye to all of you. And thank you to all of you for having a patient hearing.
Thank you, Dr. Bhatt.